Hello, everyone, and welcome. My name is Jenny Ekholm. I'm a senior scientist working within the Human Biomedical Strategic Marketing team here at PacBio, and I want to welcome everyone to the PacBio webinar series. With today's topic, we're going to focus on how PacBio long-read sequencing is being incorporated for rare Mendelian diseases. It is my pleasure to introduce today's speakers who are going to be lending their expertise to the discussion. First up is Kristen Sund, a clinical fellow in the Division of Human Genetics at Cincinnati Children's Hospital Medical Center. And Aaron Wenger, who is a principal scientist here at PacBio. We have a lot of great material to cover today, and the presentation portion of the webinar will be followed by a questions and answers session. You're welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee dashboard. We will be recording this webinar and making it available for download in the next few days. I also want to point out that we have provided you with some useful and relevant sequencing handout that you can download from your attendee dashboard. During the course of the webinar, we will be seeking your feedback on 3 different polling questions. So with that, let's get started with the first poll before Aaron set to start his presentation. I'm going to give you a few seconds to answer the poll and then discuss the results. So thank you all for participating. It looks like 58% of you are actively engaged in rare and Mendelian disease studies, and some are not, but some might be planning later this year. With that, I'm going to hand over the presentation rights to Aaron, and he can get started. Aaron, welcome.
Hi, thank you, Jenny. Thank you, everyone, for joining the webinar today. I'm going to disable my webcam for the presentation, but I'll be back for the question and answer section. And as Jenny said, today we're going to be talking about applications of PacBio long-read sequencing to increasing the solve rate for rare Mendelian diseases. There's a nice review in the New England Journal of Medicine last year discussing the 3 key aspects of studies to associate genetic diseases with the underlying molecular cause, and named that the 3 key aspects were: first, comprehensive detection of variants, and that you can't associate a variant with the disease if you don't first see that variant; then accurate estimate of the allele frequency of a variant in the population; and then finally, an understanding of how that variation affects gene expression. And it's this first aspect of comprehensive variant discovery that's most relevant to PacBio long-read sequencing. Human variation occurs on many scales, so there are single nucleotide differences, small insertion and deletion variants, and then larger variants called structural variants, which are operationally defined as differences between genomes that involve 50 or more base pairs. And structural variants come in many different types. There are deletions, which are losses of sequence; insertions, which are gains of sequence; inversions, and translocations. And while by count there are many more small variants than large variants, when you look at the aggregate number of base pairs impacted, it's actually these larger variants that explain most of the base pair difference between any 2 human individuals. And what we've seen over the last few decades is that the ability to detect this variation has increased with technology, and so has the ability to tie genetic variation to genetic disease. So this, of course, started with visualization of chromosomes in karyotypes, which could identify chromosomal abnormalities. And for certain diseases, and this particular number I'm quoting here is from a cohort of individuals with intellectual disability, for around 5% of cases, a chromosomal abnormality could explain the disease manifestation. The main wave after karyotype was the development of microarrays, specifically array comparative genomic hybridization to look at copy number variants with a resolution down to around 50,000 base pairs, and the ability to see this the smaller scale of variation provided a diagnosis for a larger class of, or a larger fraction of individuals. For a similar intellectual disability cohort, or ACGH is able to identify a cause for around 12% of cases. And this was then followed by short read sequencing, first applied at large scale in exome sequencing studies. And these exome studies were able to identify small variation, mostly single nucleotide variants and indels in the coding regions of the genome. And this survey of much more variation in the genome brought with it a large boost in the solve rate for rare disease cases. Again, for an intellectual disability cohort, exome sequencing identifies a cause in around a quarter of cases. And then finally, moving beyond exome, there's whole genome sequencing, which is able to identify the small variants in both the coding regions and non-coding regions of the genome, and also some structural variants. And again, this increased ability to detect variants has brought with it an increased solve rate. So the general trend has been technology has enabled increased variant detection, and the ability to see more variants has increased the solve rate for rare disease cases. But importantly, even so-called whole genome sequencing with short reads is not a comprehensive assay, and it misses many variants in the human genome. In particular, short read sequencing misses small variants, the single nucleotide variants and indels, in difficult to map regions of the genome. And then it misses many large structural variants. In fact, often more than half of structural variants are missed, particularly long insertions and then variants in repetitive regions of the genome. This poor sensitivity of short reads for structural variants has been established in a number of studies over the last 5 years. And most of these studies have used long read sequencing to detect the variation that was missed with short reads. In particular, they've been using PacBio smart sequencing. The key attributes of this technology are that it generates long reads, which are on the order of tens of kilobases, and that while individual reads have errors, the errors occur randomly and the consensus accuracy is extremely high. It's also a technology that reads single molecules, individual DNA molecules, not clusters of molecules. There's extremely uniform coverage of the genome with little bias in terms of GC percentage or sequence complexity. So it's able to sequence regions with very high GC content or tandem repetitive regions. And then finally, in addition to determining the sequence of a DNA molecule, it provides simultaneous information on epigenetic marks like methylation. And the first application of these long reads to studying human genomes, as I said, revealed a lot of large variation that had been missed with short reads. But because the long reads used in these studies had lower individual read accuracy, it was difficult to detect the small variants that were seen with short reads. So it provided a very complementary picture where you would see with the long reads variation that's missed with short reads, but correspondingly there was variation seen with short reads that was missed with the long reads. So there's this paradigm where you had either short, accurate reads that were good at detecting small variants, or longer, less accurate reads that were good for detecting large variants. But last year, we introduced an extension to PacBio sequencing to generate what are called HiFi reads, which are both long and accurate. So these are reads that have a length of 20,000 bases or more, but similar accuracy, 99% or higher, with an average of 99.8%. So similar accuracy to what you see with short reads. So with this class of variation, you can get the advantages of long reads and short reads in a single data type. Just to give a sense of what a HiFi read looks like, this is selected as kind of a representative HiFi read, which has a median length and a median accuracy. And so you see here the 19,820 bases from this HiFi read, and the sequencer predicts that this read has an accuracy of Q33, or one error every few thousand bases. And that of course is a prediction of the sequencer, and then you can actually take this read and compare it to the genome and see when the read differs from the genome, which are places of potential error, and those are highlighted here in the with the red triangles. So you can see in this 19,812 base pair read, there are only 8 errors. As you see, long stretches of thousands of bases without any error, and a very small number of errors for such a long read. So this read has a 99.96% accuracy. Because these reads are so accurate, they can be applied in standard bioinformatics pipelines that may be familiar to those of you who have used short read sequencing. So starting with the HiFi reads, you can align them to a reference genome and then call variants using tools like GATK or deep variant. And this produces a VCF file, which can be phased and then visualized in tools like IGV. And this accurate and long read data type provides now a comprehensive picture of variation in the genome. So you can see single nucleotide variants, indels, and structural variants, all with these HiFi reads. And to actually measure precisely how accurate the results are, we typically use the NIST genome in a bottle benchmark. And this is an authoritative characterization of a human genome from the US National Institute of Standards and Technology. So they provide standard materials, so this is standard DNA, and a list of where that DNA differs from the human reference genome. And you can use it to measure precision and recall of variant calls. And what we see when doing that is that with HiFi reads, you have extremely high precision and recall for single nucleotide variants and indels and structural variants at 30-fold coverage. And performance remains very good even down to lower coverage levels like 15-fold. Just to give some examples of the type of variation that's seen with HiFi reads, here's one example of a structural variant. You see here on the top PacBio HiFi reads and on the bottom short reads for the same human individual. And with the HiFi reads, you can see a 1,733 base pair deletion that's homozygous. And this variant is obscured in the short reads by mapping to a repetitive region of the genome. One other thing to note in the HiFi reads is that these colored tick marks denote single nucleotide variants, and the reads tend to be long enough that there are multiple heterozygous variants that can be contained in a single read, which thus allows you to connect those variants into haplotypes, and you can separate reads into the maternally inherited and a paternally inherited genome. So you see here the two piles of reads in the top of those in haplotype 1 and those in haplotype 2. You can see that the deletion is present in both haplotypes, thus being a homozygous event. The second main class of variation that's missed with short reads but seen with HiFi reads is small variants in difficult to map regions of the genome. And here's an example of such a region. It's a few exons of a disease-causing gene, STRC. This is a congenital deafness gene, and this portion of the gene, the box portion of the gene, has a pseudogene copy in the human genome that is very similar in base content, and thus it's difficult to map short reads correctly to the functional copy of the gene or the pseudogene. However, the PacBio HiFi reads are long enough to span through this repetitive region and map uniquely to the functional copy of the gene, and you can see all these single nucleotide variants that are clearly revealed with the HiFi reads that are missed with short reads. And this gene is just one of many medically relevant genes where HiFi reads provide additional mappability. So this table shows a list of 193 medically relevant genes that are known to be problematic for short read sequencing. And for 152 of those, HiFi reads fully resolve the genes. And for the remainder, they provide some additional information. So going back to the paradigm I discussed at the beginning, we now are at a point where long read sequencing, particularly with HiFi reads, can reveal single nucleotide variants, indels, and structural variants. And this increase in the ability to detect variation, if trends follow from the past, will bring with it an increase in solve rate for rare disease cases. And that's what we have started to see. So the first success with the application of long read sequencing to a Mendelian disease case was published in 2017. And that was this case of an individual who presented at 7 years old with a tumor in his heart. Over the next 10 years, he appeared with a number of different tumors in glands and recurrences in the heart. And at the age of 18 was sent for a genetic screen and the geneticist diagnosed him with a disorder called Carney complex based on the phenotypic presentation. But molecular testing for the known Carney complex gene came back negative, and there was no molecular explanation for the disorder. A few years later, this individual had short-read whole genome sequencing to try to get a molecular explanation for the disease, but again, that came back negative. And then finally, a few years after that, he was sent for long-read sequencing on the PacBio sequel system to screen structural variants. And in this genome, the PacBio sequencing revealed around 15,000 variants, large insertions and deletions, and these variants were then filtered to focus on those that were most likely to be an explanation of disease, eliminating those that were common in the population, and then zooming in on the ones that affected coding exons, and focusing first on the ones that affected coding exons of known disease genes. And that left a list of 6 candidate genes. A manual curation of these revealed heterozygous deletion of the first coding exon of this gene PRKARA, which was the known Carney complex gene, but where the previous assays were unable to see this this deletion event. So here you're seeing a pile up of PacBio reads, and half of those reads show a 2.2 kb deletion that knocks out the first exon of PRKARA. Sanger sequencing of the parents of this individual showed that this was a de novo event, and RNA sequencing confirmed that the expression of PRKARA was half of the wild type level in this individual. Thus, this variant was classified as pathogenic and causative of the disorder in that individual. So this case ended with a molecular diagnosis being given through PacBio sequencing of a large deletion. Since this case, there have been many other examples of the application of PacBio sequencing to identify disease-causing variants in Mendelian cases. Here showing a similar story, but now rather than a de novo autosomal dominant disease, this was a presumed autosomal recessive disorder. There were 2 siblings who presented with myoclonic epilepsy, but where exome sequencing failed to identify any causative variant. One of these individuals had PacBio whole genome sequencing, and that revealed a homozygous deletion of the first exon of the gene CLN6. And again, CLN6 was a known myoclonic epilepsy gene, but this first exon has extremely high GC content and thus is difficult to sequence with short read sequencing. And that obscured the fact that the lack of reads in this exon was not, or it made it difficult to differentiate a technical artifact from biological variation, although that biological variation is very clear with the PacBio sequencing. The group that performed this study then suggested as a general framework that people consider for rare disease cases, if you have a strong candidate gene, starting with a targeted assay. If you have multiple candidate genes or no single strong candidate, starting with a whole genome or exome short read sequencing assay. And then if those assays are uninformative, to reflex to PacBio whole genome sequencing to be able to get at this hidden repeat expansion of structural variant variation. Since that time, with the development of HiFi reads, an alternative strategy is to start directly when you have multiple candidate genes or no strong candidate gene with PacBio whole genome sequencing to look at not only the structural variants, but also at the small variants. And we've seen many groups applying this approach. There are now a few review articles talking about the disease-causing variants that have been discovered with long-read sequencing, and we'll hear some more about that from Kristen a little later in this webinar. So to come back to this again, now with long-read sequencing, it's been well established that there's additional genetic variation that's discovered with HiFi reads, and now a number of examples of how that additional variation brings with it an increased solve rate for rare disease cases. One outstanding question, though, is exactly what that solve rate will be, and there are some larger efforts now to start answering that question. In particular, there's a consortium funded by the European Union called SolveRD, which is looking at applying PacBio sequencing to 500 human individuals to start answering the question about exactly what % of those cases that have been left unsolved by short-read whole genome sequencing will be solved by long reads. And similarly here in the United States, Discovery Life Sciences and at the Hudson Alpha Institute for Biotechnology has started to apply long-read sequencing to thousands of human genomes to characterize both the common and disease-causing variation. In terms of the technical roadmap, PacBio is working on making HiFi reads higher throughput and more affordable, with a target by the end of this year of enabling one human genome to be sequenced on a single run with one SMRT Cell in one day, and again with this one data type 1 sequencing technology. So with that, I'll hold for questions at the end and turn it back over to Jenny.
Thank you, Aaron. With that, I think we're going to launch our next poll. So you're going to see a question on your screen. For rare and Mendelian disease studies at your organization or in your lab, what is the estimated solve rate currently? And the choices are less than 25%, between 25 and 40%, between 40 and 50%, and greater than 50%. I'll give you a few seconds to answer. Great, thank you. The most common answer for 51% answered between 25 and 40%. With that, I will hand over the presentation to Kristen Sund, who will talk about how they used long-read sequencing in 2 of their neurological undiagnosed disease cases.
Hello there, can you see me okay?
Not yet, so hold on a moment.
Now?
Yes, we're still waiting for your screen though. One moment. Let's see, do you have the presentation, presenter rights? So you need to launch your presentation still.
The presentation is up.
Are you sharing your screen?
Yes.
Oh, there we go. Perfect.
Okay. Good afternoon, everyone, and thank you for joining me today as I talk about the use of long-read technologies for the diagnosis of neurologic disease. My name is Kristen Sund, and I am a certified genetic counselor, and I am finishing up as the first combined Laboratory for Genetics and Genomics Fellow at Cincinnati Children's Hospital. I am going to go ahead and disable my video cam so you can see my slides.
Kristen, can you, yeah, presenter mode, thank you.
Yep. Cincinnati Children's is located in the Midwest in the southern part of the state of Ohio. It is also known as the Queen City or the city that sings. Cincinnati Children's Hospital has more than 15,000 employees and is ranked in the top 3 pediatric hospitals in the United States. Our Division of Human Genetics Diagnostic Laboratory currently has more than 90 employees and runs a variety of tests ranging from traditional chromosome analysis to exome sequencing. The Diagnostic Laboratory hosts the American Board of Medical Genetics and Genomics LGG Fellowship, and that is where this work was performed. Although the first human genome was sequenced in 2003, we have only recently begun to appreciate the complexity that is built in by structural variation. As technology improves, with new instruments like those used in this study, the PacBio Sequel and the Bionano Sapphire, we are finding that the number of structural variants is in the range of tens of thousands per genome, a dramatic change from the early days of microarray when studies showed 221 copy number variations in 20 individuals. With these new tools, we can begin to find clinical utility for testing human samples. Long-read technologies have been used to detect and characterize highly repetitive and GC-rich regions, including triplet repeat disorders, highly polymorphic regions, to discriminate between a coding gene and a pseudogene, and to identify retrotransposon insertions. These tools are also being used to better characterize structural variation, like copy number variations and chromosome rearrangements. For example, Stansu et al. Performed extensive mapping and phasing of structural variants to define the breakpoints of patients with complex chromosome rearrangements, but did not define etiology. Chasson et al. Performed extensive mapping and phasing of structural variation with multiple platforms and multiple softwares in 3 trios to set a gold standard for structural variation using multiple complementary laboratory techniques. Although a number of studies used long-range sequencing to characterize a clinically relevant copy number variation, only a few have focused on defining the genomic content at the breakpoints of a translocation, including the paper by Dutta et al., which showed disruption of the RGAF9 gene in intellectual disability. We set out to understand how these new tools may help find an answer for some of our undiagnosed patients. We included patients with a neurologic disease who had a known chromosomal rearrangement, but no known gene cause for their phenotype. Patients also had additional previous genetic testing that was normal or non-diagnostic. The goal was to use low-pass, long-read SMART sequencing to obtain base pair resolution of the breakpoints in the rearrangements in order to identify a gene that was responsible for their phenotype. And this is the story of the 2 patients we were lucky enough to meet along the way. Our first patient is a 9-year-old with short stature and hypotonia. She came to our attention because she had nocturnal epilepsy that was initially difficult to control. She also had developmental delay and speech delay and was observed to have some self-stimulatory behaviors as seen in the video. There was no family history of another individual with the same features. Previous testing included a normal SNP microarray and a normal epilepsy next-generation sequencing panel, which revealed a variant with unclear clinical significance. I first worked on the patient's chromosomes in 2017, and we reported a karyotype with multiple breaks due to a translocation between the long arm of chromosome 2 and the long arm of chromosome 4, an insertion of genomic material from the long arm of chromosome 2 into the short arm of chromosome 4. Even before we determined that the entire rearrangement was de novo, I was convinced that the gene could be found at one of the breaks, so we enrolled her in a research study. With help from the Structural Variation Grant from Pacific Biosciences and the University of Minnesota Genomics Center, I was able to perform low-pass whole genome sequencing on the patient. We obtained average genome coverage of about 8x with 4 SMART cells. The average read length was 10,633 base pairs. The patient had 31,324 structural variants, of which 168 were BNDs that passed quality control. In this data, we found 6 breakpoints representing 3 events. We were able to confirm all 3 breakpoints with PCR and Sanger sequencing. However, there was a gap between the confirmed sequence breakpoints from the translocation, and we could not find evidence of the insertion based on default PBSV processing. Importantly, the break on the derivative 2 happened in intron 1 of the MBD5 gene. MBD5 stands for methyl CpG binding domain. The protein belongs to a family that includes MECP2. These proteins are known to regulate gene transcription through chromatin remodeling. Importantly, haploinsufficiency for this gene causes autosomal dominant MBD5-associated neurodevelopmental disorder, with clinical features of developmental delay, hypotonia, severe speech impairment, seizures, sleep disturbances, and autistic-like behaviors. Although this was a close match for the child's phenotype, a literature search revealed multiple isoforms of MBD5 and showed that exon 1 was generally non-coding. Thankfully, in this case, the gene is broadly expressed, including in whole blood. We were able to get a fresh sample and extract RNA, then perform quantitative reverse transcriptase PCR to show that there was up to a 2- to 4-fold reduction of expression, consistent with a disease mechanism of haploinsufficiency. In addition, we found evidence that previous patients have been reported with deletion of exon 1, resulting in haploinsufficiency. Although our patient did not have a deletion of exon 1, the translocation of exon 1 to another chromosome would be expected to result in a similar consequence. Importantly, this gene had been sequenced on the epilepsy next-generation sequencing panel and was normal. All of this was supportive evidence, but a return to the patient phenotype put it over the top. When I met the family to get a sample, I noticed a distinct hand posturing in the patient that had also been documented in her chart. In a review of the literature, such stereotypic and repetitive behaviors, especially hand movements, were commonly reported in these patients. So I thought we had an answer for the patient, but I still had questions. As I mentioned, we were able to Sanger confirm the derivative 2, and we even learned something about mechanism. This figure shows the actual breakpoint on the derivative 2, with chromosome 2 at the top and chromosome 4 at the bottom. Sanger sequencing revealed a deletion of 32 base pairs at this breakpoint and some microhomology, which may have led to non-allelic homologous recombination. Though we were able to find the break on the derivative 2, we could not PCR the expected complementary break on the derivative 4, despite multiple attempts with different primer pairs. In addition, there was a centromeric break-in that could not be explained, so I began to suspect that something more complicated was happening. With the help of Bionano, we were able to obtain optical mapping data, and even at the level of the Circos plot, we could tell that it was more complex than we suspected. On the derivative 2, we saw one break and were able to confirm that it fell within the MBD5 gene. Things were a bit more complicated on the derivative 4, and we began to understand why we were unable to confirm the complementary 4q breakpoint, which would contain the rest of the MBD5 gene. This is just one of the images from the optical mapping data, but it shows that the break on the Q arm was not as we expected. In addition to the translocation, there was a large pericentromeric inversion indicated by the large teal bracket. Likely prior to the large inversion, there was an insertion of the long arm of chromosome 2 into the long arm of chromosome 4 next to a smaller inversion, which is denoted by a smaller teal bracket and adjacent to the purple 2q insertion. We are still working through some additional complexity. We were able to use the combined data to map and characterize all of the breakpoints. Not surprisingly, most of the breakpoints were associated with highly repetitive elements. In addition, other genes were present at the identified breakpoints and were broken apart, but were not considered candidates because they did not match the phenotype or were not associated with strong disease-causing evidence, were the wrong kind of variant, and/or had low constraint. For the breaks that were not initially identified by long-read sequencing, a return to the BAM file and a close inspection of the subreads found representative reads even though they were not in the process VCF. These often overlapped with subreads calling the complementary break. This is still a work in progress, and there are ongoing PCRs and Sanger sequencing happening in the lab to finalize the structure of the derivative 4 and better understand the mechanism behind each break. The family has given consent to receive research results. There is no existing clinical long-read test to confirm the genomic findings. There are some labs that could confirm the genomic finding with another technology, but at the end of the day, that does not confirm disease in this patient. I did find a lab that has clinical testing for MBD5 RNA expression, and I think this will best address the patient needs and clinically confirm the diagnosis. This family has been looking for an explanation for their daughter's epilepsy for 9 years. In addition to having an answer for their daughter and resources about the condition, her prognosis, and clinical management, they will also be able to use this information to connect with other families who have a similar diagnosis. Moving on, patient 2 has a movement disorder. This is an area that I am highly interested in and also an area of interest for long-read sequencing, as 2 new genes have been identified in this population through long-read studies. Both were repeat disorders. Our patient is a 17-year-old with chorea, myoclonus, anxiety, and hypothyroidism. She had a family history. Her dad also had a movement disorder, and he passed away in his early 40s due to lung cancer, though he was a non-smoker. Previous genetic testing included abnormal chromosomes with a small but visible paracentromeric inversion on chromosome 14. The patient also had a normal microarray and normal single gene testing for NKX2-1, which is found on chromosome 14. Targeted sequencing of the NKX2-1 gene, also known as the TITF1 thyroid transcription factor, made perfect sense as a candidate because it is involved in the development of the thyroid, the lungs, and the forebrain. Pathogenic variants in this gene cause autosomal dominant hereditary benign chorea, which in addition to a movement disorder causes hypothyroidism and lung disease. Additionally, there are reports in the literature that this gene is associated with early death due to chronic lung disease or pulmonary adenocarcinoma. This is consistent with the findings in the patient's father, who also carried the inversion. All of this fits very well with the patient and the family history, but no coding changes in NKX2-1 were identified. We suspected we might find an intronic break in this family. Long-read sequencing was performed and obtained an average genome coverage of about 8x, and the average read length was 10,436 base pairs. The patient had 29,241 variants, of which 141 were BNDs that passed quality control. In this data, we found 8 breakpoints representing 4 events, but 2 of them appeared to be overlapping, so we set out to confirm 3 breakpoints and were able to do that. We quickly saw that none of the breakpoints disrupted the NKX2-1 gene, but there were a couple of breaks that were close. One possibility for disease mechanism was a change in gene regulation, so it became more important than ever to understand the genomic landscape in this region to determine disease mechanism, and I was having difficulty doing that based on breakpoints alone. Again, we were able to combine the long-read data with the Bionano optical mapping data to determine that there were actually 5 breaks and to reassemble chromosome 14 in the patient. It turned out to be more complex than suspected, almost a mini chromothripsis event. We found that the region containing NKX2-1, highlighted with an asterisk over segment D, was inverted and inserted in a completely different regulatory environment. In this case, long-read sequencing called 4 out of 5 breakpoints, but again, as indicated on the bottom right of this screen, we were able to return to the long-read BAM data to pull reads that were representative of the missing BND. This base pair resolution is important for our clinical assessment to know what genes are at the breakpoints and to indicate the presence of deletions or duplications at the breakpoint. It is also extremely useful for primer design. Again, we found a number of repetitive elements at the breakpoint and no other candidate genes, and we set out to understand how this rearrangement could result in the disease. This figure represents the landscape surrounding NKX2-1 on chromosome 14. The dark black dotted line on the right indicates the location of NKX2-1, and the green lines show the location of the breakpoints in this patient. To date, A variety of mutation types have been reported, including missense, nonsense, splicing, indels, and gross deletions. Deletions typically overlap the entire NKX 2-1 gene and often include surrounding genes. However, at least 2 cases have been reported where deletions did not include NKX 2-1, but were upstream of the gene and were thought to be responsible for the unique phenotype. Those are indicated here with horizontal blue lines. Based on these cases, it was initially thought that the MBIP gene may be responsible for a phenotype similar to autosomal dominant hereditary benign chorea, but no independent pathogenic variants were identified in the gene, and there was speculation that the phenotype could result from dysregulation of the NKX 2-1 gene. Our case is the first case to show that translocation of the upstream region with MBIP intact results in hereditary benign chorea. Additional work is needed to confirm this. Unfortunately, NKX 2-1 is not expressed in the blood, and it is difficult to obtain the sample types needed for this work. At the moment, we are focused on Sanger confirmation of the newly identified breakpoints and finalization of structure and mechanism. We were able to collect PBMCs on this patient, and they could be used to create induced pluripotent stem cells and differentiate them into relevant neuron types for various experiments. With the appropriate controls, these differentiated neurons could be used to evaluate for decreased expression of NKX 2-1 and its targets. In addition, HiFi could be used to determine how changes in the physical genome lead to disruption of the regulatory environment and dysregulation of the NKX 2-1 gene. In summary, in both cases of neurologic disease, a combination of chromosome analysis, long-read sequencing, and optical mapping data identified breakpoints and elucidated a causative gene. Neither long molecule technology alone called all the breakpoints at base pair resolution at this depth of coverage. Identified candidate genes had been previously sequenced in the patients by earlier generation technologies, and no causative variant was identified. Breaks occurred in the non-coding sequence of the gene. Quantitative RT-PCR was able to confirm decreased gene expression in 1 case. Sanger sequencing confirmed breakpoints and helped determine mechanism. In addition to the microhomology identified, both patients had small inversions near a breakpoint, which may lead to genomic instability and predispose to structural variation. Sequencing of healthy parents to better understand their genomic structures may help us to better understand this predisposition on a research basis. Both characterized rearrangements were more complex than predicted. Although this is a small sample size, it does lead to speculation that most or all quote-unquote simple rearrangements are actually mini chromothripsis events with multiple breaks and rearrangements. In all cases, break-ends not called by the default PBSV pipeline were present in the subreads in the BAM file for long-read sequencing. This suggests that alternative software parameters or increased coverage would allow base pair resolution of all the breaks. The use of long molecule technologies in patients with known chromosomal abnormalities is high yield because it allows for targeted analysis of long-read data. In addition, as the price comes down and the resources for filtering improve, the use of this technology for most types and sizes of structural variation can be implemented. This application in the clinic will be used for diagnosis, prognosis, management, and treatment of patients with genetic disease. This application in undiagnosed patients will be incredibly meaningful to families who have been searching for a diagnosis. So what are the clinical laboratory implications? Some minor points are that this testing requires a lot of DNA and unique DNA extraction protocols. Even the 2 long molecule technologies I worked with in this study required different extraction techniques to enable the extraction of diverse product lengths. But more importantly, and more related to application of the technology, analysis of structural variation should not be limited to coding regions. Supplemental testing is required to show that breaks in non-coding sequence result in dysregulation of gene expression to support disease mechanism. Sample type is a consideration as not all genes are expressed in whole blood. Where possible, it is important to identify all breaks of a structural variation to rule out other candidate genes. The identified complexity of structural variants in these patients suggests multiple breaks can be found in most patients with structural variation and increases the chances of an incidental finding at one of the breakpoints. Reflex to long read or optical mapping clinical testing following a normal panel allows for targeted analysis of long read data, may increase the diagnostic yield of a given panel, and may also increase the proportion of probands with a detectable pathogenic variant in known disease-causing genes. Most importantly, I would like to thank the patients and their families for participating in this research. I would also like to thank Matt Hestand and Lisa Dyer, my mentors for this project, Teresa Smolarek and Brian Dawson, who are the leadership of the Laboratory for Genetics and Genomics Laboratory. This work couldn't have been done without the support of my neurology colleagues, Dr. Barb Hallinan and Dr. Stephen Wu. I received a lot of additional support from colleagues at Cincinnati Children's, PacBio, University of Minnesota Genomics Center, and Bionano Genomics, and they are listed here as well. Thank you for listening, and please reach out to me with the email on this page if you have any questions or comments. And with that, I will turn it back to Jenny.
Thank you, Kristen, and thank you for everyone for submitting numerous questions. Before we start the Q&A session, I want to take a few seconds for our last poll. So you will see a question on your screen now. How many active unsolved cases do you have that could potentially benefit from long-read sequencing? Select one of the following: none, 1 to 5, 5 to 10, 15 to 20, or greater than 20. Thank you. And majority landed greater than 20, and then it's a spread across all of them. But with that, I will take it into the Q&A session. Thank you again for submitting your questions. Please feel free to add additional questions as well once we get started. The first question is for Aaron. How do you decide on a study design needed for different cases? For example, trio versus singleton cases, and how much coverage would you recommend?
Sure. So I think here in terms of trio versus singleton, I think the considerations are the same as with exome sequencing. So I think certainly if there's a suspicion that it's an autosomal dominant disorder, then having the trio is fantastic, so you can confirm the candidate events being de novo. Even for recessive, of course, it's great to see that one variant is inherited from the mother and one from the father. So in general, I think we would recommend, just as with exomes, that trios will give you power when available, but you can proceed with singletons when trios aren't available. In terms of coverage, I think our standard recommendation is to go with 15x HiFi read coverage per sample. There is a small increase in indel calling performance at higher coverage levels. So I think for the highest sensitivity, you can go to 30x coverage, but our standard recommendation is 15x coverage. I think one thing to point out is Kristen's results actually used the previous instrument that PacBio sold. So that was all done on the Sequel system, which had a lower throughput than our current system, the Sequel 2. So now I think Kristen mentioned doing around 8x coverage per sample. And I think today that was really a compromise between the cost of doing the assay and the power for discovery. But today, I think that would be done at a higher coverage. So 15x would be our standard recommendation.
Thank you. Let me see, there's a question for Kristen. Is there data from the parents on the MBD5 patient? Are all the findings de novo?
Yeah, so we were actually able, the parents had not initially had chromosome testing, but we were able to, during the time of this research study, we were able to get them to go back and they were clinically tested, and the rearrangement was completely de novo. So we have not actually sequenced them for this gene, but there's no reason to believe that since they don't have a chromosome rearrangement that they would have a break in this gene.
Thank you. For Aaron, are there any genetic variation that makes some populations more sensitive to infectious disease than other as a result of single nucleotide polymorphisms?
This is outside my expertise, but I would say certainly there's genetic variation that predisposes different groups to different disorders, and I'm sure the same goes for infectious disease. I think maybe one thing we could point to is actually if you go to the PacBio website now, there's a banner focusing on the work on using PacBio sequencing to study COVID-19, and there's some I think references there talking about variation, particularly in the major histocompatibility complex, that can be characterized very well with PacBio sequencing that may correlate with susceptibility to that disease, and I'm sure it's similar for other diseases.
Okay, thank you. For Kristen, did you say your patients were sequenced with CCS/HiFi reads or with low-fold long reads?
Yeah, so I do believe it was the HiFi reads, but it was, as Aaron said, not on the latest machine. So we were just on the Sequel, and they are now up to the Sequel 2.
Thank you. And for Aaron, how much difficulty with long read sequence analysis is presented by deficiencies of reference sequence? Can a quasi-independent assembly be carried out that ensures that the regions missing in the reference are subject to analysis?
Sure. So if I understand that question, one limitation of standard variant calling approaches is that they involve taking reads and mapping them to the reference genome. So it's actually an advantage of long reads in which if there's say 6,000 or 8,000 bases in an individual that's not in the reference genome, those bases may be just impossible to map with short reads. But with long reads, you can, if you have a 20,000 base pair read, you can anchor on the left and right and see where these novel 6 or 8,000 base pair 8,000 bases are in the genome. However, there is some limitation to that. So if the individual has, say, 1 million bases that aren't in the reference genome, then that's not going to be contained in a single read. And that's the type of thing that you may be able to get with alternative analysis approaches that don't rely on mapping to a reference genome. So you could, I think as the questioner asked, consider doing de novo assembly of the reads. So here you don't use a reference genome at all, you just compare the reads to each other and try to reconstruct the chromosomes. And then you can take those reconstructed sequences and try to interpret them. That is, I think, an exciting analytical direction to look at. But it is a more complicated process, and I think the most of the value for structural variants can be found in the standard reference-based mapping approach. But long term, I think that the idea of doing de novo assemblies in an unbiased way is the kind of right long-term vision, and that there will be things you see with that you don't see with reference-based mapping.
Thank you. For Kristen, when you say going back to BAM for the unresolved breakpoints, is this looking at read mapping adjacent breakpoints or chimeric fusion reads that map to two distant genomic locations?
Yeah, that's a really good question. So when I initially started doing that, I was using the breakpoints that I got from Bionano, which are not at the level of a base pair resolution. So initially I would put in those breakpoints, and I often had to zoom out pretty far to actually see the subread of interest. And often that subread wound up mapping to a region where we already had a break. So it was the complementary end of another break. And since my coverage was lower, I would, maybe there would only be 1 or 2 subreads that represented this and it wasn't being picked up by PBSV. But basically what I would do is just click on every individual subbreed until I got to one that looked like it was correct. And then we went back and verified those breakpoints with PCR and Sanger.
Thank you. If we, for Aaron, if we did variant calling, no, sorry. I'm not sure who this is for. If we did variant calling for the non-HiFi PacBio technology, does this mean we need to rerun the sample with the HiFi platform?
I would say no. The ability of the PacBio sequence to generate HiFi reads doesn't eliminate the utility of the long reads. So I think if you've already generated sequencing data and done analysis, that analysis is still valid. And in particular, if you're focusing on structural variants, I think you can identify those with high precision recall with the kind of the traditional long reads. The advantage of doing the HiFi reads is you'll be able to also find the smaller variation with high precision recall.
Thank you. For Kristen, how many patients have Cincinnati Hospital used this technique for?
So we're just getting started with it. So I actually applied for this funding about 1.5 years ago, and these were our first 3 patients as far as I know.
Okay. Thank you. For Aaron, is PacBio developing any software tools to call repeat expansion variations?
Yes, so I guess maybe I should throw this one to you, Jenny, and it's an area you work on. But so in general, the PBSV software that Kristen used for calling structural variants is able to find repeat expansion variants, although they are not annotated as such. So they will be seen, if a repeat expands, it would be called as an insertion of repetitive sequence, but then there are some additional software tools that we have that are focused on specifically repeat expansion disorders. And we can, I can send to this questioner a link to those, but there's in particular a target enrichment technique called NOAMP, which lets you pull down specifically repeat expansion loci for sequencing on PacBio. And the analysis pipeline for that NOAMP enrichment technique is focused on repeat expansions.
Thank you. And for Kristen, have you considered RNA sequencing to complement your whole genome sequencing?
Absolutely. I think it would be fantastic. And I've even thought about that in terms of the patient where we were able to perform quantitative RT-PCR and we were able to confirm that there was decreased expression of MBD5. The place that offers clinical testing can either do an individual gene or can do basically a transcriptome, a clinical transcriptome. And I've gone back and forth on what to do, but to me, there is actually some information about the targets of MBD5 that there's, it's a smaller study that was done, but does show some upregulation and downregulation of different genes. And so it almost seems worthwhile to have the transcriptome done and be able to see not only that you have haploinsufficiency for MBD5, but also that its targets are being dysregulated.
Okay, thank you. For Aaron, in the novel study designs that incorporate PacBio to solve multigenic or unknown genetic causes, what is the recommended output or coverage? For short reads, there are a number of recommendations from different institutes, for example, 100x or 150x coverage.
Yeah, so I think the standard recommendation, as I said before, would be 15x coverage with HiFi reads per sample, and then for the high sensitivity, 30x coverage, but 15x is the standard recommendation.
For Kristen, using both optical and smart sequencing would seem very expensive. Will cases like Kristen's be solvable by smart sequencing alone in the future?
I hope so. I think it would be nice if we could do everything in one test. You know, I was lucky enough to get grants and get help in having the sequencing done, but I know that's not practical in the long run. And so I think, you know, what I saw from looking directly at the data is that I can pull these reads out. And if you think about the fact that I really only had an average of 8x coverage it's not too surprising that that PBSV was not able to pick up some of the reads. That said, I think one thing that's important to point out is knowing the reads by itself isn't enough, especially for cases like the NKX2-1 case, where I really wanted to know what the landscape itself looked like, and it was very difficult for me to do that by just looking at the breakpoints. So it was helpful there to have the optical mapping to be able to put the chromosome pieces back together.
For Aaron, for detection of SNPs, indels, CNVs, and structure variants, what standard pipeline would you recommend? It seems that for CNV/structure variants, there is no quote-unquote standard pipeline like GATK.
Yeah, so for the HiFi reads, the recommended pipeline for structure variants is PBSV, which is the kind of PacBio provided pipeline. There are some nice academic pipelines as well, but PBSV is the one we recommend, and that will find structure variants and copy number variants. And then for small variants, we recommend Google Deep Variant. You can also use GATK, but the performance is significantly better with Deep Variant, so that's what we recommend.
Okay. For Kristen, what are the inclusion criteria that you accept for including patients in your studies?
So this was really just a pilot for us. Specifically for the pilot, I'm very interested in neurologic disease, so I was looking for patients that had a known chromosome rearrangement, but no known underlying gene, and they had to have some neurologic phenotype. They also had previous genetic testing that was normal or non-diagnostic. So they had, in all cases, they had a normal SNP microarray, and both of them had some type of sequencing that would have covered the genes of interest, but still they had those tests were negative.
Thank you. For Aaron, is the analysis of PacBio similar to that of short reads, i.e., do I need other software to analyze it, and can I follow the same pipeline?
So I think the answer is that yes, it is similar, but that there are different software packages that would be recommended. So the basic framework is still map reads to a reference genome, and then that produces a BAM file, and then take that BAM file and run it through a variant color, which will give you a VCF. This is the same pipeline that everyone is accustomed to, but for mapping reads, while many people use BWA for short reads, we recommend Minimap2 or a wrapper for that called PBMM2 for PacBio reads. This is actually by the same author as BWA, so Hang Lee wrote both BWA and Minimap2, and the usage is very similar, but it is a different piece of software. And then for calling variants, again, we recommend Google Deep Variant for the small variants, and then PBSV for the structural variants. And these software have very simple usage, just like you're accustomed to for other things. You give it the alignment file, the reference genome, and it gives you VCF.
I think this is for Kristen. Any variations you observed in sequence analysis by previous methods and this long read data?
I'm sorry, I'm not sure I got the question.
And I was too quick to remove it from the dashboard. Okay, so let's move over to the next one. I apologize for that. Any questions that we have not been able to answer here, we will follow up in an email. Have you ever done testing for patients with normal karyotype without a candidate gene region or a gene? And if so, how are the results?
Yeah, I have not done that, but I really want to, because I think that there are a number of cases. So if you think about gene reviews, you go in there, you look at your gene of interest, and it will usually lay out for you, this is the condition, this is the phenotype, these are the percentage of patients that have a mutation in NF1, for example. But usually there's a missing percentage, and there are a couple of possibilities with that percentage. One is that those patients actually don't have NF1, right? Another possibility, and what I expect that we will see moving forward in this area, is that they have structural variations that cannot be detected by microarray and cannot be detected by next generation sequencing. I can't give you an exact percent, but I definitely think those cases are out there, and I'm really excited about these undiagnosed programs that are applying long read technologies, because I think that will give us a better idea how many of those cases can actually be solved by long read technology.
Thank you. We have quite a lot of questions, but I'm going to ask one last question. And again, for the ones that we didn't get to, we will answer you over email. Is it possible to go for targeted smart sequencing when we need to know the parental origin and imprinting status of, say, a case of an UPLD? I don't know. So basically, can we do a targeted?
Maybe I can answer that.
Yeah.
Great. Maybe I can answer that one. So yes, you can. I think the only trick there is exactly how you target the region. So I think standard ways like PCR, if you can generate it, you can sequence it on PacBio. You can do long range PCRs to PCR 10 kb molecules or longer and sequence those on PacBio. And then there are also some non-PCR enrichment techniques if required. I think you saw in talk did a follow Sanger sequencing, and I think depending on exactly what you want to see, the PacBio sequencing will give you very high throughput. So if you just want to look at one region and one sample, it's probably overkill to sequence 100 gigabases, but
We're losing you there slightly. All right, thank you for your answer. With that, I'd like to thank Kristen and Aaron for their time today and sharing their expertise and perspectives on long-read sequencing. In closing, I want to mention briefly 3 things. So first of all, many of you asked if this session is recorded. Yes, it is. And recording will be available for download within the next several days. So keep an eye out for a follow-up email with a link to the recording. Secondly, to stay up to date on our upcoming webinars, such as this one, visit and bookmark pacb.com/events. And finally, immediately following this webinar, you will receive a brief questionnaire. Please take a few minutes to fill it out. It's just for us to develop, make sure that we develop the right content for future webinars that interest you. So again, thanks for joining us today, and I hope you join us all again in the future webinars. And with that, take care and have a wonderful day.
Thank you.